biotechnology from Jim Hyderabad in 2016. Dr. Raju Yadavalli has completed PG diploma in patents law with A plus grade from Nalsar University, Hyderabad in the year 2015. He has won Young Engineer Award 2013 to 14 in Chemical Engineering discipline from Institute of Engineers, India. She has 21 research publications, including 5 book chapters. She has published a patent on microbial pigment composition as nucleic acid staining agent that was published in the year 2022 along with other inventors. She received travel grant by UGC, Government of India, twice for research paper presentations at Australia and the UK. She is nominated as advisory member for implementation of Skill Weekend program, Department of Biotechnology, GOI, at state level by Telangana State Council of Science and Technology in 2021. She is also a lifetime member of Biotech Research Society of India. She is the project coordinator for establishment of ATAL Community Innovation Center at CBIT for encouraging startups funded by ATAL Innovation Mission of a budget of worth rupees 2.25 crores. She is also the director of the SPV company established for setting up ACIC CDIT. She is having 18 years of experience teaching and research. Her research interests are bioprocess engineering, algal fuels, bioreactors design, and alternative energy sources. Dr. Rajshri secured a research project from Department of Biotechnology, Government of India on microalgal biomass production in energy efficient internally illuminated bubble column photobioreactor for biodiesel production worth rupees 33.82 lakhs. She is undertaking a consultancy project to develop a device for capturing carbon dioxide from the atmosphere for a period of 6 months from Neon Lake Enterprises Private Limited. We are very honored to have you here with us, ma'am. With immense pleasure, I would like to call upon Dr. Rajshri Yadavalli to enlighten us with her knowledge on energy and exergy analysis and carbon sequestration. Over to you, ma'am.
Thank you so much for your kind introduction. I really thank Dr. Gopinath Garu, Dr. A. Sai Ramesh, and the student who has given an introduction about my research, whatever little I have done all these years. Thank you so much for that. And today, this talk will be divided into 2 sections. In the first topic, I'll be concentrating on microalgae and its value-added products. And in the second part, I'll be concentrating on the carbon dioxide sequestration in a biological method, how it can be done better, and what are the various available methods for the carbon dioxide sequestration. So, can I share the screen? Am I audible? Is there any disturbance in my voice?
You are well audible, ma'am. Please go ahead, ma'am.
Yeah. Hope you are able to view my screen.
Yes, ma'am. Yes, ma'am.
Okay, thank you. So, coming to the topic, I was actually asked to talk about exergy analysis. Let me just brief you about what is exergy. Exergy analysis is nothing but a thermodynamic analysis, which is primarily based on second law of thermodynamics. Energy analysis, what we know usually, that is based on first law of thermodynamics. However, this exergy is based on second law of thermodynamics, which involves assessing and comparing processes and systems Actually, this is about exergy, and here I would like to relate my topic to exergy in a way where we are assessing and comparing various processes of microalgal cultivation for production of biodiesel and value added products through biorefinery approach. So all these years, from the past 12 to 14 years, I have been into this microalgal biotechnology as my specialization. I have worked on several microalgal species. At the beginning of my PhD, my objective was to um, optimize the biomass from microalgae and use it as a source for biodiesel production. With that motto, I have started my PhD, and with the leads I got in my doctoral studies, I have extended my work. And uh, as I completed my PhD, I secured a DBT project, a government project of worth 33.82 lakhs approximately, where I could actually build a bubble column photobioreactor, which I claim it as an energy efficient bubble column photobioreactor for optimization of biomass. So I'll be focusing on this 12 years of journey, especially on the microalgae part. I will be concentrating on this particular in this particular talk. So um, this will be the contents for this talk. Introduction, how to enhance the lipid productivity, what strategies I have applied, how can we integrate the wastewater treatment along with the microalgae, and what value added products we have focused on from this microalgal cultivation, and a conclusion. So first of all of you must be knowing, or all of you must have heard the term microalgae, which is buzzing these days. Microalgae is nothing but a photosynthetic microorganism. It's just like a plant, but it is a microorganism which requires sunlight, water, CO2, and some nutrients for its growth. So these days, carbon neutral fuels are gaining lot of popularity in view of the global warming. Even my next topic is very much related to this global warming. And what is actually meant by carbon neutral fuel? Neutral fuel means they neither contribute or they will not reduce the amount of carbon in the atmosphere. So biofuels, whatever biofuels you consider, like bioethanol, biodiesel, biobutanol, all these are considered as carbon neutral fuels. And whatever I am going to talk now, it is a biodiesel has various sources. You might be knowing that Jetrofy previously was used as a major source of biodiesel production. Now, what we have done as part of my research study, it is a production of biodiesel from microalgae. So microalgae, they are characterized by the production of significant amounts of biomass and simultaneous lipid production, which can be used as a feedstock for carbon neutral fuel. And also, microalgae is a very much potential source of renewable energy. Additionally, microalgae can also be used to produce bioethanol, biofertilizer, various value added products, cosmetic products, animal food. Like this, microalgae has several benefits. It extract the end product out of the microalgae. Now, let me show a as I explained you, microalgae is a photosynthetic microorganism. So it requires sunlight, water, CO2, and at the same time it requires very being a microorganism, any microorganism requires nutrient for its growth. So the nutrient can be provided either by an artificial medium. The artificial medium which we have optimized for the growth of microalgae is BG11. It is a standard medium, but however we have optimized certain parameters based on the local species we have isolated. So apart from the artificial medium, even the food waste, sewage wastewater, industrial wastewater, heavy metals, flue gas. All this also can serve as nutrient source for the microalgae. So we'll focus on one of these items also in our research. So this is how the microalgae grows, by taking all the inputs. And if you see the output from microalgae is capable of producing carbohydrates, which might lead to 3G fuels. It is capable of producing lipids will in turn convert into biodiesel. And simultaneously, along with lipids, what we have discovered, or what we have come to know from literature studies and even from our research, is that it is capable of producing pigments which are of highly medicinal value, like astaxanthin, lutein, flavonoids. All these can be characterized as nutraceuticals. So simultaneous production of lipids and pigments is possible in microalgae. And biomass is also one of the product of microalgae, which can be used as biofertilizer. Apart from all this, even microalgae is being used in MFCs for bioelectricity production. So this is just an overview of what microalgae can do by taking the inputs. So, I'm jumping directly into my research. Kindly excuse me. I know research will be very much narrowed down to our specializations, which might be difficult for others to understand, but I'll try my best to explain what I have done during my research journey. So, the expert actually, what I am focusing here is the major experiments which has given a lead for us to jump into another experiment. Such experiments only I am focusing in this particular topic. If any one of you are interested to conduct research in microalgal biotechnology, please listen to all this carefully, because this will definitely help you in starting your work. You can start off, because we have already reached a certain level. If you take it from that level, definitely you can reduce the time, and also um, um, you can find out some new things which earlier was not found out by the scientists who are working in the area of microalgal biotechnology. So the first topic, what we have discovered, everyone knows microalgae is capable of producing biomass. It can produce biodiesel, okay, but there are certain limitations. What limitations means? If biomass is produced in more quantity, lipid content will reduce. Indirectly, it is affecting the biodiesel production. So it's not just either biomass or lipid. Previously, in 1900s, when this microalgal research has been started, it is just like whether we want biomass or lipid. But after certain optimization techniques, the research scientists have came to a conclusion that obviously they are inversely proportional to each other. However, we can optimize the conditions so as to give better quantity as well as quality. Quantity in terms of biomass, quality in terms of the lipid content of the microalgae. So, in this experiment, we were interested actually to know how the microalgae will be influenced by the uh, transition in the modes. There are actually three modes of operating any microalgae: autotrophic mode, mixotrophic mode, and heterotrophic mode. Autotrophic mode means, in autotrophic mode, we will not be giving any carbon supplement to the medium in which we are growing the microalgae. However, the inorganic carbon in the form of sunlight or artificial light, whatever we are providing for the microalgae to grow, that serves as the carbon source, which will help in the growth of microalgae. That is about autotrophic mode. Coming to mixotrophic mode, the second mode of cultivation. In mixotrophic mode, we'll be giving organic carbon as a supplement and dissolved inorganic carbon as well. Like suppose we can give glucose in the medium, as well as some dissolved inorganic carbon source what we have used here in our experiment is NaHCO3. So these are supplemented in addition to the sunlight or artificial light is provided for the microorganism. In addition to that, we are giving some supplementation of carbon in the medium as well, and this is called as mixotrophic mode. And in heterotrophic mode, we are neither giving inorganic carbon or organic carbon in medium, or we are maintaining dark conditions for the microalgae to grow. So this is about how actually we operate autotrophic, heterotrophic and mixotrophic modes. And in this experiment what we have done is we have taken uh, 3 flasks with mixotrophic mode, 1 flask with autotrophic mode, 1 with heterotrophic mode. We have grown these cultures in growth phase for 9 days. The candidate species for our cultivation is Chlorella pyrenoidosa. So we know its growth curve, and based on its growth curve, we understood that growth phase can be maintained for 9 days. And after 9 days, we have created stress phase. Stress phase was intentionally created so that all of you please know that in stress phase, microalgae accumulates lot of lipids. So stress phase means either low light intensity or nitrogen starvation conditions or high temperatures. Depending on our end product, we can create the stress conditions. Here in our experiment, the stress conditions are low light intensity and nitrogen starvation. First, we have not given any stress and we have allowed the microalgae to grow. Later, we have created the stress conditions, and also we have changed the modes from mixotrophic to autotrophic, mixotrophic to heterotrophic, mixotrophic to mixotrophic. We wanted to see how this trophic transition induces the lipid productivity inside the microalgae. Similarly, autotrophic to mixotrophic and heterotrophic to mixotrophic. After growing again for 9 more days in stress phase, we have harvested the biomass, dried the biomass, and the lipid being intracellular inside the microalgae, we have to disrupt the cell. And there are various optimization techniques which we have employed for disrupting the cell as well. And we have found that probe sonicator is the best method to disrupt the cell. So we have opted this cell disruption using probe sonicator to extract the lipids out of the biomass. And the extracted lipid was quantified using Blick-Dyer method, and then we have transesterified using acid catalyzed transesterification. And then whatever lipid we have obtained, we have analyzed for fame using gas chromatograph. So this is a results isotrophic to mixotrophic mode. Biomass content was high when there is isotrophic mode, both in growth phase and stress phase. However, there is a little decrease in stress phase, the biomass, which could be attributed to the nitrogen depletion because we have maintained the nitrogen starvation conditions. And if you see this particular slide, productivity of lipid was highest in mixotrophic to mixotrophic mode. It was 284 grams per kg, which was followed by mixotrophic to heterotrophic mode, which is 266 gram per kg. And similarly 243 grams per kg in mixotrophic to autotrophic mode. So this why we analyzed why it could be the reason. So then we found out that it might be attributed to the partitioning of excess carbon from photosynthesis into the lipid synthesis pathway, whatever we have provided. So the maximum total lipid productivity was in mm means first letter stands for growth phase, second letter stands for stress phase. Similarly, MH and MA modes. And also we have analyzed what is a neutral lipid productivity, because neutral lipid productivity is the one which is actually contributing for the biodiesel production. So we also analyzed the neutral lipid productivity, and we have observed that the neutral lipid productivity was again higher in mode when compared to other modes. And these are some graphs we have obtained. And so from this experiment, what we concluded is, if we maintain mixotrophic mode in both growth phase and stress phase for this chlorella pyrenoidosa, we could get more lipid content as well as more biodiesel content. This is the lead, which you can note it down even if you are not very much familiar understanding all these values and all. But this is the conclusion of this particular experiment. Then coming to optimization of stress conditions in chlorella sorokiniana for enhanced lipid and PUFA content. PUFA content is the one which is responsible for the quality biodiesel production. So we have to definitely, when we do gas chromatograph, we will be analyzing PUFA content only. So now we wanted to optimize the stress conditions by for another species, that is previous species was chlorella pyrenoidosa, and this species is chlorella sorokiniana. Here we wanted to enhance the lipid and PUFA content in this particular species by optimizing the stress conditions. And this is the pathway which you all know, the biochemical pathway. So from this biochemical pathway, always biochemical pathway will help us in understanding what are the precursors, what are the inhibitors, which can help us in increasing our desired product. So what we have identified is stress factors such as nitrates, sodium thiosulfate, which is actually the inhibitor of this Krebs cycle, can help us in optimizing the lipid content for chlorella sorokiniana. So for optimizing these parameters, we have chosen Plackett-Berman experiment. Plackett-Berman experiment, it is one of the statistical tool which will help us in understanding the factors which are responsible, which are highly influencing that particular yield of the product, yield or the product. So the best results whatever we have optimized from this Plackett-Berman design, they were further considered to conduct the experiments at larger scale for the lipid production. So here what we have done is, again we have divided the cultivation into two phases, growth phase and stress phase. In growth phase, we have taken 4 flasks of chlorella sorokiniana. Of course we will be taking duplicates to ensure the results, the same results, sustained results. So along with duplicates, we have taken 4 flasks which were actually grown in modified BG11 medium. This is the artificial medium. We are not using any wastewater here. That was actually done in the later part, which I'll be explaining you. So along with the external carbon supplementation, that means we have operated in mixotrophic mode, and the stress phase was induced by adding different carbon sources. We have added pyruvate, malate, acetate and glucose in stress phase as different carbon sources for the microalgae to grow. And here we have analyzed what is the unsaturated fatty acid percentage for various carbon sources we have given in the stress phase. And what we have observed was, at the end of growth phase, because there is no different carbon source that is used, uniformly the lipid percentage was in an average of 12%. However, in stress phase, the lipid percentage has increased, and the lipid percentage was more in pyruvate. Unsaturated fatty acid percentage was more in pyruvate. However, the final lipid percentage was more in glucose. see the difference. This is overall lipid percentage. Again, when we have done the FAME analysis, we understood that unsaturated fatty acids percentage was in this proportion. And even though the lipid percentage was highest in glucose with 56%, the unsaturated fatty acid percentage was highest in pyruvate. It is not bad result that glucose even got a better, it stands second in all these four with unsaturated fatty acid percentage of 67.4. So what we have concluded from this particular experiment, these are the gas chromatograph graphs with chlorella soriciniana malate as the carbon source in stress phase, pyruvate as the carbon source in stress phase, acetate, and glucose. As I have shown you in the previous table, these are the unsaturated percentages. Pyruvate has yielded more unsaturated fatty acid percentage. However, the lipid percentage was more with glucose. And even in this unsaturated fatty acid percentage, which was 74.21, or all the chlorella soriciniana, whatever might be the glucose source, in all the conditions, 16 is to 1, 18 is to 3 are the most commonly observed unsaturated fatty acids. Even we have, this is the most important, interesting conclusion what we have got was heavy carbon chain fatty acids were also observed. 20 is to 0, 20 is to 1, all these were also observed, especially 20 is to 5 also we have observed, which is actually a very important food supplement. With this lead, what we have concluded was, so if we can modify the modes and if we can induce some stress phase with various carbon sources, we can even get DHA, EPA, which are of very much interest for today's world. Even if you apply for government grants for some projects and all, this particular EPA and DHA, they are focusing a lot. That's why, so this lead can help you in getting into such kind of productions even from microalgae, because microalgae are also capable of producing EPA, DHA. So the next experiment was on as I told you previously, only we handled artificial medium. Now I wanted to test how the wastewater actually helps in the growth of microalgae, and that's why we have taken a continuous stirrer tank reactor which is of 5 liters capacity. And in Hyderabad we have taken an area called Uppal, where there is a highly polluted lake. We call it as Nalla Chervu Lake. So from that water is used, that sewage water is used as a medium. Of course, we didn't use it directly. We have actually taken the different dilutions of sewage wastewater, and we have even worked out which dilution is giving better growth of microalgae. So we have taken the sewage wastewater and we have started growing our microalgae. And at the end of the growth or stress phase, we have, you can see at the beginning it was just looking like sewage wastewater, but at the end microalgae has grown very well in the sewage water. So sewage water, the flow is like this. We have collected the wastewater, we have analyzed what is the various parameters, what are the parameters, BOD, COD, pH, all nitrates, phosphates, everything we have analyzed for the wastewater. And then we have grown our microalgae in continuous stirrer tank reactor, and which can actually serve as treatment for this wastewater. And then we have harvested the biomass, analyzed the treated water, and even the biomass that is harvested, which is separated from the treated wastewater, is used as biofertilizer. And for this treated water, we have checked the survival assay of Danny or Aereo fish as well. And this is the actual, of course, very much related. This is the topic which I have presented at Elsevier conference in USA. So this is the overall work what we have done, and I'll now show the results as well. So these are the initial concentrations of the sewage wastewater and the final concentrations after treating the wastewater, and these are the removal efficiencies, which you can observe the removal efficiency is much better at a this is an energy efficient process, that is what we are claiming actually. So if you take the survival percentage of the fish also, the survival percentage was 100%. That means the wastewater initially was highly toxic for 24 hour, 48, 72, and 96 hour days. But after treating the water and when we have grown the fish, it was all the fish was 100% alive, and we claim that water is completely safe for the aquatic environment, the treated water after completing the algal growth. So even we have tested the microalgal biomass which is harvested after the growth in the CSTR. So without biofertilizer and with biofertilizer, we have created sample fields in some plastic tubs, and we have analyzed the growth. We have done this experiment several times to ensure the results, and we have uh, got these interesting results. With and without biofertilizer, there was a percentage increase of up to 30% in the root length and 20% in the shoot length. So we thought definitely, even if we go for some wastewater treatment as well, the biomass which is being obtained can be used as a biofertilizer. This is one more conclusion. However, here I would like to highlight one point. Whatever we are using, see, microalgae is a suspended culture. Uh, and harvesting the microalgae itself takes lots of time and energy, and lot of biomass in this process might get wasted. So what we have done here is we have immobilized the microalgal cells instead of using the suspended culture. We have actually taken the microalgae, immobilized them, just normal immobilization process, sodium alginate, calcium chloride, the same process we have adapted, and then we have put the beads in the reactor. These are the beads inside the reactor, and we have even checked the bead stability. This is the 0th day bead, and this is the 18th day bead. See how much green it has turned into. That means microalgae is immobilized inside the bead. Why we have immobilized this microalgae is the only reason, harvesting. Because already we are going with wastewater treatment, we cannot invest more money in the harvesting of algae, because that is the actual bottleneck for any microalgae process to be commercialized at a larger scale. The harvesting is a very costlier process, apart from the cultivation. So we wanted to reduce the cost by immobilization. When we immobilize it, obviously separating it from the treated water is very easy. Just by normal filter cloth also we can separate these microalgal beads. That is how we wanted to reduce the cost. So this proposal, actually we have proposed it to Telangana Science and a science department, where just beside the lake we wanted to establish an artificial pond with microalgae. This microalgae then will go into a settling tank. After the growth period, we'll be harvesting in the settling tank. The biomass will be settled down. That biomass will be sun dried and it will be used as biofertilizer, and whatever water is treated, that will be again sent into the lake, which will be actually safe for the aquatic environment, or it can be even used for irrigation purposes. This is our proposal at a pilot scale. This is about the next experiment. Then coming to value added products. what we have tried to extract from our microalgal species. So we have worked on some products like lutein, astaxanthin, flavonoids. Here I am not highlighting about lutein, because that work is not yet published. Whatever I am showing now, all these works are published in various reputed journals. So, coming to the extraction of flavonoids from microalgal pure strains, that here the strains we have taken are Chlorella vulgaris and Chlorella pyrenoidosa. We have adopted a biorefinery approach. For extracting these flavonoids from Chlorella vulgaris and pyrenoidosa. Flavonoids, as you know, are a class of plant and fungus secondary metabolites, and they are even reported in microalgae. And again, when we have I suggest all of you to please go through the biochemical pathway of the microorganism, whatever you are considering. When you take the biochemical pathway, you will understand what is a precursor which is responsible for enhancing such end product, whatever we want. And when we have analyzed, we found out that L-phenylalanine in the medium composition definitely will affect the flavonoids production in the microalgae. So we have taken it as a precursor and we have added it in the medium composition. We have modified the medium and we have grown the microalgae. So this is the method we have adapted. Media we have prepared, then microalgae, these two microalgae we have added, and again we have adapted two different modes, autotrophic mode and mixotrophic mode. Then we have added L-phenylalanine in two modes, and then after growth period we have harvested the biomass, then extracted the flavonoids. And after extracting the flavonoids, this is the actual beauty of the microalgae. It has the capacity for simultaneous production of flavonoids as well as lipids. It is not that either flavonoids will be produced or lipids will be produced. Both can be extracted. So we have first extracted the flavonoids and then we have extracted the lipids from biomass. And after extracting the already we have standardized the procedure for lipids using RGC and all. So the flavonoids also we have confirmed using various assays, and these are the results we have obtained. Dry cell weight, how it was varying with CV stands for Chlorella vulgaris, CV in autotrophic mode, CV in mixotrophic mode, CP stands for Chlorella pyrenoidosa in autotrophic mode when it was grown, and Chlorella pyrenoidosa when mixotrophic mode it was grown. And when you observe, CV mixotrophic mode has given us maximum dry cell weight of up to 14 grams per liter. So after this we have even analyzed what is the internal carbohydrate composition, total flavonoid content in different modes, and lipid percentages also in different modes we have analyzed. So the highest dry cell weights, as I told you, it was obtained in CV autotrophic mode, followed by CV mixotrophic mode. So Chlorella vulgaris is obviously the candidate species which is giving us more biomass when compared to Chlorella pyrenoidosa in whatever mode it is. So uh, also we have observed highest lipid percentages in mixotrophic modes of both CV and CP, which were 11 and 9 percentages. Highest carbohydrate content was observed in case of CV mixotrophic mode, followed by CV autotrophic mode. Similarly, the highest amount of flavonoids were obtained in case of CP autotrophic, even though the biomass was more in CV mixotrophic. The highest flavonoid was obtained in case of CP autotrophic mode at day 18, followed by CP mixotrophic mode at day 16. We have analyzed day-wise flavonoids as well. And then we have obtained maximum flavonoid content in CV mixotrophic mode and CV autotrophic mode. And these are the HPLs. we have confirmed the presence of flavonoids using HPLC analysis, and this is the conclusion of this particular experiment. We have got some interesting conclusions again. Flavonoids and chlorophyll content were high in Chlorella pyrenoidosa. At the same time, nitrates were maximum consumed by CP autotrophic mode, and we have analyzed a relation between this nitrate consumption and flavonoid content. Though carbohydrates were increasing as the days of cultivation were increasing among the four, that is CP autotrophic, CP mixotrophic, CV autotrophic, and CV mixotrophic, CP autotrophic mode had least internal carbohydrates, and higher amount of lipid percentages were observed in Chlorella vulgaris. Thus, we concluded that flavonoid synthesis is somewhere linked with nitrates and carbohydrates. As more amounts of nitrates are consumed, flavonoid synthesis was increasing. That is what we have observed, and this can be taken as one of the important lead. Then coming to I think this is the last experiment which I'll be focusing in this particular talk. So, we have even conducted some comparative studies on simultaneous production of lipids and astaxanthin from Chlorella sericiniana in a two-stage operated photobioreactor. We have conducted this particular experiment in 5-liter airlift bioreactor. Previous experiment, whatever I have shown with respect to wastewater treatment, was conducted in continuous stirred tank reactor. This particular experiment we have conducted in two-stage operated airlift bioreactor, which we have customized and we have made it in our lab. In this investigation, we have evaluated biomass and lipid productivity of Chlorella sericiniana in again two phases. Again, we have observed the biochemical pathway, and we have understood that FeSO4 4H2O, if it is added along with sodium acetate in the medium, that too in the stress phase, it will induce astaxanthin. Hope you must have listened about astaxanthin, which is a carotenoid which is of high importance these days as a nutraceutical. It costs a lot. Usually, whatever astaxanthin that is being produced in markets is produced in a chemical way, not in a biological way. Whatever we are focusing is a biological way, and the candidate species generally chosen for astaxanthin production is Haematococcus pluvius, which grows at very high temperatures in stress phase. That's why Haematococcus pluvius is generally grown in desert regions. You must have studied this if you have interest in this microalgae. So here we wanted to even understand the potential of Chlorella sericiniana, because we came across some papers where they have focused on extracting these astaxanthins from various Chlorella species. Then we thought, why not Chlorella sericiniana? So we have operated our airlift bioreactor in two phases: growth phase and accumulation phase, or stress phase. Lipid accumulation phase or stress phase. In stress phase, what we have given is, as I told you, by understanding the biochemical pathway, we understood that FeSO4 4H2O along with sodium acetate, if it is given, astaxanthin might increase inside the microalgae. So after the lipid extraction, we have even analyzed for astaxanthin presence using HPLC analysis, lipid analysis, gas chromatograph, and these are the results we have obtained. We have even studied the radical scavenging activity using this H2O2 assay. And these are the fatty acid profile assays, and these are the HPLC chromatograms of the species we have taken. So the results, whatever, this is the airlift bioreactor what I am talking about. Astaxanthin is a red color pigment. That's why we have obtained the pigment in this color. So the results indicated that the supplementation of FeSO4 2H2O, inorganic carbon, and nitrate effectively manipulated the production of astaxanthin in Chlorella sericiniana. We have confirmed the astaxanthin presence using UV visible spectroscopy. The wave scan we have monitored, and and even the HPLC, which we have outsourced this work, and we have obtained the HPLC results. The relationship between lipid and astaxanthin production, this CS, Chlorella sorokiniana, what I'm talking, is a freshwater algae, and this relation is also confirmed using scavenging activity. Maximum yield of biomass was 11.2 grams per liter and 5.1 grams per liter at the end of growth and stress phase. I told you we have operated in two stages, and the lipids were analyzed using gas chromatograph, and we have found that unsaturated fatty acids were much higher than saturated fatty acids, which is actually desirable for us when we are going for biodiesel production at a commercial scale. And unsaturated fatty acids like palmitoleic acid or the linoleic acid, EPA, all these were present. And this astaxanthin being a powerful antioxidant, it is also showing radical scavenging activity, which was assessed by H2O2 assay, which I have shown, and the maximum radical scavenging activity was 91%. And this comes the last one, sorry. So we've also wanted to see how a fruit extract can help in growing the microalgae. Till now, whatever we have grown was in a closed system, not an open system. So here in this experiment, we have taken grape maize and we have diluted it in different concentrations, and we have grown microalgae. This is the open pond which is growing microalgae with grape maize fruit extract, and this is the control. So we have designed this small scale raceway ponds, what we claim it as. Here in control, we have used BG11 as the medium, and here grape maize was used as a medium, and both were grown in fed batch mode. At regular intervals, we have analyzed the composition of the media, and we were adding the feed at regular intervals, and harvesting of biomass was done every 3 days after attaining the exponential phase. And then we have estimated the lipids and carbohydrates. Why carbohydrates means we wanted to use this particular microalgae as a source for bioethanol production. So even we have analyzed carbohydrates apart from the lipids, whatever we have done. And this is a mixotrophic mode of cultivation. This capacity is 40 liters. So we have operated this, and we have compared the pH values in fruit waste and control. This is the comparison of dry cell weight in fruit waste and control. Carbohydrates, how they are distributed in grape pomace and control. And if you see, carbohydrates are more obviously in fruit waste, and internal carbohydrates of fruit waste and control. If you see this grape pomace, it was decreasing, increasing, decreasing, increasing. This is because it is operated in fed batch mode. Whenever it was decreasing, again we ensured that sufficient amount of grape pomace is added in the feed, so that the internal carbohydrate concentration is again increasing. And even we have estimated the chlorophyll to understand how the photosynthesis is going inside the microalgae for both fruit waste and the control. And biomass and lipid productivities were analyzed on day 10 and day 12 for grape pomace and control. So the maximum biomass was achieved in mixotrophic mode, which was 1.9 gram per liter for BG11 medium. And in the fruit waste, it was 1.5 grams per liter. The internal carbohydrates, as I told you, in the test sample was highest than in the control. This was obvious. We expected the same because we were using the fruit waste as a supplementation in the test. And the ml, and it was one, whereas it was 1.7 microgram per ml in the fruit pomace. So the lipid content was estimated on day 10 and day 12, and it was observed that highest lipid percentage was in test sample. 23% was there in test sample when compared to control sample. And this is the fatty acid profile we have obtained for day 10 and day 12 for our grape pomace as well as the control sample. So We have concluded that the Chlorella sorokiniana was cultivated in small scale ponds in beach-11 and grape pomace. Various analyses were performed, and we have observed maximum lipid and carbohydrates were produced on day 12 in test sample. And lipid profiling revealed 17 is to 1, 18 is to 2 in this particular experiment. And these are my students, and this is the project which I have got, under which we have worked on all these experiments. Thank you so much for this opportunity. I think I have taken, I am will be continuing about carbon dioxide sequestration, but before that, I would like to know if you have any doubts in this particular session. Sir? Can you hear me, sir?
Ma'am, am I
Yeah,
audible, ma'am?
yeah, you are audible.
Yeah, okay. Thank you, ma'am. Thank you so much for a clear and a detailed explanation on the research work that you're doing.
Mm.
I'm Krishna Veni from Department of Microbiology, PSG College of Arts and Science, Coimbatore.
Yeah, please, ma'am. Yeah.
Yes,
Thank
madam.
you so much.
Ma'am, very interesting that you were able to try with different, applications on algae with astaxanthin, with grape pomace, etc.
Yeah.
Ma'am, I have one doubt is how far you could scale this up. You said you were able to do a raceway pond, get the pond water and treat it with a microalgae, and then let it back into the pond and the treated water back into the pond. So, what was the quantity, volume that you had tested doing this, madam?
Ma'am, we didn't get an opportunity because being lake water, which is a public source, we had to take government approval. However, the scale, whatever we have operated, was still 40 liters in an open pond. And in a closed pond, we have operated till 15 liters, ma'am.
Okay, ma'am.
Yeah, but once we get proper approvals from government, because this cannot be done at our convenience, we have to take proper approval from the state government, and then I think we can scale it up to a bigger level. My plan is actually, if the pond capacity is some 40,000 liters or 50,000 liters, at least 10% of that should be taken as an artificial pond with microalgae should grow, and it should be finally taken to a harvesting unit where the biomass will be extracted. That is our goal. Yeah.
Yes. So that is what I wanted to understand, if what was the volume that you could take it for.
Yeah. Yes. Thank you so much.
Thank you, ma'am. Thank you.
Any other questions?
Good evening, ma'am. This is Karunya from VTHT College.
Yeah.
Ma'am, you have just studied the survival of the zebra.
Yeah. You are not audible. I think there is some internet issue with you or me. I don't know.
Zebrafish.
Arunya ma'am, there is the internet problem on your side. Can you please check? Ma'am?
Can you hear me now?
Yeah, it's fine now.
Ma'am, actually, you have studied the survival assay of zebrafish, right, ma'am?
Yeah.
But what about its behavioral aspect? Does the microalgae affect its behavioral aspect?
We didn't actually study about the behavioral aspects, but we have studied in various resources, that even in the papers that are published or articles, microalgae is being used as a food supplement for aquaculture. Like all the fish or prawns, whatever they're growing, this is being used as a feed. Definitely, I don't think there will be some major behavioral aspects when algae is being fed to the fish. Am I answering your question?
Yes, ma'am.
Yeah. That is what we have understood, but in general, we frankly speaking, we didn't study the behavioral aspects of the zebrafish when they are grown in the treated water. But never we have fed the microalgae to the fish. That is for sure.
Okay, ma'am.
What we have done is just we have separated the microalgae from the treated water, and in the treated water we have seen how it is growing. That's all. Of course, we have given
Okay.
artificial food for that, not the microalgae, whatever has been obtained as a biomass.
Okay, ma'am.
Yeah.
Actually, ma'am, one more question is there.
Yeah.
You have used microalgae biomass as the fertilizer to grow plants.
Yeah.
Does the microalgae affect the pH of the soil?
It affects the pH of the soil, ma'am. We need to
Okay.
actually, yeah, it affects the pH of the soil. And right now, we are actually experimenting on the NPK values, how the soil is being affected when this biofertilizer is being given. We are doing right now that particular study, so I cannot confirm about anything. But it will affect the pH of the soil. So we need to take some alternative measures when microalgae is being used.
Okay, ma'am. Thank
Yeah.
you so much, ma'am.
Yeah.
Participants, if you have any other queries, you can feel free to unmute yourself and you can discuss with the ma'am. Otherwise, we will give you a short break. Ma'am will take a 5 to 10 minutes break, and thereafter she will join with us for the second session.
Yeah.
Ma'am, you can take a 5 to 10 minutes break.
Thank you, sir.
And then you can join us the second session, ma'am.
Sure, sir.
So participants, there are kind of short notice about the food and hospitality and transportations. So the response so far I have received around 20 numbers only. So I think many of you maybe forgotten to fill out the Google form which I shared in the yesterday's meeting, as well as to posted the same content in the our FDP WhatsApp group also. So those who are not completed yet, so you can fill out the form so that we will come to know that the your requirement about the accommodation as well as the transportations, so that we will effectively serve with you without any hassle. So I request every one of you, please spare the sometimes to fill out the form. And if you have any queries on your mind, so feel free to discuss with me. There is no issues. We want to give you a better platform, both whatever the your travel as well as accommodation, and also we want to you provide you good lunch. This 5 days definitely uh, this will make you like some kind of memories on your mind. So yeah, such a kind of FDP that kind of feedback only we are expecting from you all. So hence I request you all to spare some time to fill out the forms. Hi sir, Nagendra Reddy sir, how are you sir?
Hello, sir. Good evening, sir. I'm fine, sir.
Yeah,
You, sir.
good evening, sir. I think tomorrow is your session.
Yes, sir. Yes, sir. No, I just wanted to listen to our
Madam,
seniors
yeah.
talk. So, yes, sir.
Okay, I got it.
Yes, sir.
Yeah.
So tomorrow, I'll be.
Yeah, exactly the same time only. So around 6:45, I think all participants will join the meet. So by 7 exactly, we will start the program. And
Done, sir. Done, sir.
maybe you can
I'll
take
be available.
bit break between the two sessions. No worries on.
Sir. Okay. Done, sir. Done, sir.
Yeah. Thank you, sir.
Thank you. Thank you.
Anything point to be need to communicate to me from the participant sides? Ma'am, one more session is there, ma'am. At the end of the second session, I will post the attendance, the feedback link in the chat room, ma'am. Meera, ma'am. Thank you, ma'am. Any other points you need to, communicate to us? So you can feel free to communicate us so that we will effectively serve for you all. Few of them not daily updated their attendance. So I am actually taking everyone's presence in the Google window. Based on that only, I am marking the attendance. So in the same time, if you fill out the form also, that will give the 100% assurance to mark the attendance in effectively in the ATAL, AICT web portal. Yes, ma'am. Jaya, ma'am. Jaya, ma'am, your raised hand. Any queries?
Shall we start the session, sir?
Yes, ma'am. Please, ma'am.
The screen is visible.
Yes, ma'am. Please, ma'am.
Welcome back, all the participants. Now, in this session, I'll be talking about carbon dioxide sequestration in general and how algae can also contribute for carbon dioxide sequestration through a biocircular economy approach. And what are we doing in this particular case for carbon dioxide sequestration? I'd like to highlight these points in this particular session. And these are the contents for this session. Introduction, then what are the various methods that are already proven for carbon dioxide capture, and what can be adopted in the current situations. I'll be highlighting all the methods, the principle wise about adsorption membrane technologies, and how algae also helps in this particular process of carbon dioxide capture. And then I'll be focusing on what is a fixation efficiency by algae, what are the various cultivation systems and the types of algae that can be used for this carbon dioxide sequestration, um, whether microalgae also can be used for carbon dioxide sequestration, what are the factors that are affecting this carbon dioxide sequestration, and what is the impact of flue gases on algal biomass, and what model we have developed. I would like to show some of the pictures what we have developed with respect to this particular concept. So coming to the introduction, you all know these days we are hearing a lot about global warming, climate change. There are a lot of concerns from all the developed countries. India is one of the major contributor for this climate change. The flue gases those are emitted out of the combustion of fossil fuels. Those are the main sources of greenhouse gases, and because 68% of the total emissions are caused by carbon dioxide, and this carbon dioxide obviously contributes to the global warming and climate change. And to add to this, it was found that increase in CO2 concentration was also due to the human activity in industrialization. The transportation, whatever we are using these days, also is a leading cause for accelerating this CO2 levels, and which in turn leads to accelerated climate change. Furthermore, the global concentration of carbon dioxide has increased from 280 ppm to 368 ppm in 200 years, uh, which necessitates actually a serious effort to develop novel technologies to reduce and mitigate these carbon dioxide emissions. So, till now in India, there is no particular commercial carbon capture and storage technology that is developed. So it has lot of potential. This particular topic has lot of potential for research as well as to get projects, or if you want to become an entrepreneur also, you can focus on this particular topic where carbon dioxide can be sequestered. So so, depending on the location where you wanted to capture this particular carbon dioxide, there are three different approaches that are being used for this carbon capture and storage system. First one is post combustion capture, pre-combustion capture and oxy-fuel combustion. Post-combustion capture means the name itself is suggesting CO2 is removed after the combustion of fuel. That means CO2 will be produced because of the combustion of the fossil fuel, and then after that we need to sequester the produced CO2. Another one is prevention is better than cure, where pre-combustion capture, which is involving separating and storing CO2 before combustion. Oxy-fuel combustion is where coal is burned in the presence of oxygen to produce a concentrated stream of CO2, which will be supplied into the system wherever you want to sequester that particular CO2. But out of all these three methods, post combustion capture, pre combustion capture, and oxy fuel combustion, post combustion capture is attracting the researchers or the entrepreneurs as well, because the other two methods, pre-combustion capture and oxy-fuel combustion, they are very very expensive, and it is difficult to implement, that too at a commercial scale. So even apart from so now in this particular topic, in this particular session, I am only focusing on post combustion capture techniques, whatever that available um, till now, whatever we know, and then what can be improved in those particular technologies, that is what we will be discussing. So from a botanical point of view, more than 50% of total photosynthesis process takes place in algae. Algae here I mean both microalgae as well as macroalgae. So algae obviously they represent a promising feedstock for CO2 mitigation with limited dependence on weather or irrigation conditions. We need not much bother about the season. Of course, it will affect the microalgal productivity, but our main goal is to sequester CO2 here, not biomass optimization or lipid optimization. Instead, the main goal in the previous lecture you must have heard, we are optimizing the stress conditions to enhance the lipid content or astaxanthin or the flavonoids, whatever we are targeting for. Here our main target is CO2 mitigation, CO2 sequestration. So as I told you, both macro and microalgae are being studied for these purposes. However, more focus is towards microalgae when compared to macroalgae, because macroalgae are not capable of producing value added products like biofuels, whatever we have discussed. If we can optimize those in macroalgae, definitely they can also be a potential feedstock for CO2 mitigation along with microalgae. And mm, researchers have found that microalgae are able to sequester 10 to 50 times more CO2 than their terrestrial counterparts. What are the terrestrial counterparts? Trees, the plants. So, be it from atmospheric air with low CO2 concentration. Hope you must be knowing how much concentration of carbon dioxide is there in atmospheric air. It amounts to up to 0.04%. In atmospheric air, the concentration of CO2 is very low. So, algae can even sequester the atmospheric low carbon dioxide concentration or from flue gases as well. Flue gases obviously contain high amount of CO2 concentrations, 12 to 15%, when compared to atmospheric air, which is at 0.04%. So, uh, microalgae are capable of sequestering any of these. So also here there is lot of necessity for conducting research which microalgae is capable of sequestering CO2, that too when it should have the tolerance for SOx, because SOx is a toxic gas for the microalgae. However, NOx can be used in its metabolic activities, metabolic process in its biochemical pathway, but SOx cannot be utilized, and it is a toxic gas for the microalgal growth. So we need to identify such species which can withstand these high levels of SOx and NOx. And also, as we have discussed earlier in the previous session, because of the high photosynthetic efficiencies, algae can accumulate biomass as well as lipid content. This lipid content can lead to biodiesel. Also, not just biodiesel, any value added product from microalgae, in addition to sequestration of CO2, will definitely help, because two birds, one shot. That's what we are targeting for. So now let us see the methods of CO2 capture. As I told you, we will be only focusing on post combustion techniques. Post combustion techniques means once the fuel is combusted, we'll be getting CO2 in the flue gas. So here in this figure, this post combustion technique is being highlighted. Although many post combustion CO2 capture technologies such as adsorption, membrane separation techniques, which we will discuss now in this lecture, all are there. None of them is actually providing an ideal solution for the flue gas that is being emitted out of the transportation or from the power plants, because of mass transfer limitations. Also, when you opt for this post combustion techniques like adsorption and membrane separation technologies, here we need to store the CO2 that is being absorbed. Here, CO2 to storage, and there is always a risk of CO2 escaping back to the atmosphere, and also transporting CO2 to the storage sites was a major concern, which is actually involving lot of costs. So absorption technique, let us focus on the post combustion absorption technique. Let us understand what is this. So this is amine based chemical absorption. There is a physical absorption technique as well, which we will discuss briefly. So this is a chemical absorption technique which is developed to absorb the CO2 or capture the CO2 in post combustion method. Here in this process, what we'll do is we'll supply the flue gas to the solvent from bottom of the absorption tank. So the solvent being monoethanolamine, MEA, that is a choice for chemical absorption always. So this will be sent into the flue gas in an absorption uh, tank, and once the absorption is completed inside that particular tank, the CO2 rich solvent, this MEA, will absorb the CO2 from the flue gas, and it will be sent into the stripper. We generally call the desorption process, wherever it occurs, we call that as stripper. And in the stripper, it whatever this solvent MEA which has absorbed the CO2 will release the CO2 again inside the stripper, and now the fresh regenerated MEA will be again sent back into the absorption tank for absorbing fresh CO2 from flue gas. This is how the process occurs, and this is the reaction which is actually showing what is happening inside the absorption. The forward reaction is taking place inside the absorption tank, and the backward reaction is taking place inside the stripper. This is also called as absorption and desorption reactions. Forward reaction represents absorption, backward reaction represents desorption. But of course, I agree this is a reversible reaction, but here when you observe, the forward reaction takes place at normal temperatures, but the reverse reaction, the desorption, whatever we are talking inside the stripper, that takes place only at higher temperatures. And when I am mentioning higher temperatures, definitely lot of energy is being involved in the process. High energy consumption is being utilized. High energy is being utilized for high temperature solvent regeneration inside the stripper. That's why This particular technology is not being commercially adopted because of the high energy costs that are involved in the stripper. Hope you have understood the principle behind this. Then coming to physical absorption, it is a method where CO2 absorption takes place without any chemical reaction like MEA absorbing CO2 and all. There is no such chemical reaction, but it is purely based on Henry's law. Henry's law, under high pressure and low temperature, the CO2 will be absorbed. Absorption is one process, desorption is the second process, right? So absorption will be done under high pressure and low temperature, and desorption will be done under low pressure and increased temperature. Again, the same problem here. High pressure, creating high pressure for flue gas is not that easy, and it again involves lot of energy consumption. Similarly, the desorption process here also involves increased temperatures. Hence this method, and of course, the partial pressure of CO2 in the flue gas is very very less, but Henry's law, this absorption process only takes place under high pressure. So this method is obviously less efficient for carrying out absorption or desorption processes, and hence absorption overall, whether it is chemical or physical, it is not being adapted for CO2 sequestration. Now let us understand what is the adsorption process. This is another way of capturing CO2 in the post combustion process. In the previous absorption process, we have used a liquid solvent MEA for the absorption of CO2. However, here we will be using solid absorbents. Solid adsorbents, why solids are preferred when compared to liquids means because of the contact area. The surface area which will be in contact with the CO2 will be more in case of solid adsorbent when compared to a liquid adsorbent like MEA. So, however, this technique again has its disadvantages like low adsorption capacities at low pressures and impact of water vapor and other gases on the adsorption, because water vapor is also present in the flue gas. 141 different compounds are present in flue gas apart from CO2. So, all this might affect the adsorption process. So many materials are proposed for this adsorption process, such as amine-based chemical adsorbents, which have been widely investigated. As I told you, obviously solid processes provide high surface area for adsorption process, but this low adsorption capacity is due to the low pressure. As I told you, CO2 partial pressure inside the flue gas is very low, and increasing its pressure only at high pressures adsorption can take place efficiently. So obviously high costs are involved. There is a need of high CO2 adsorption capacity, and hence adsorption is not successful at a commercial scale. Then coming to the third method, that is membrane technology method. Here in membrane technology method, again it is classified into two. Membranes, as you know, they offer some selectivity towards specific compounds. So here, suppose we want to absorb or capture carbon dioxide from the flue gas. So in this particular process, I'll show you with a diagram where you can understand it very easily. This is how a membrane separation process works. Membrane, it acts like a filter. Now this is the feed is nothing but our flue gas, which is consisting of several compounds. Now I want to only selectively separate CO2 from this flue gas, right? So this membrane selectively separates from our feed mixture the required gas. So during this separation process, two factors are actually influencing the process, that is selectivity of the membrane and permeability of the membrane. Membranes actually can be classified again into organic or inorganic. Organic involves polymeric membranes, inorganic involves zeolites, carbon or ceramic membranes, and it may be porous or non-porous also sometimes. So as I told you, membrane separation is characterized into two: gas separation membranes and gas adsorption membranes. Let us understand the difference between these two. See, when we are talking about biological way of sequestering CO2, why we have to go for biological way? What are the disadvantages that are associated with already existing ones? You definitely need to know. Unless and until you know the disadvantages of the current methods that are being adopted, you cannot actually justify why you have to go with a biological way of sequestering the CO2. That's why we have to understand all this. So this is gas separation membrane technique. In this, again, there is a membrane. However, at high pressure, the flue gas is being sent into the membrane, and again, because of selectivity, only CO2 will be passing through the membrane, and it will be separated out of the flue gas, and this CO2 will be captured, stored, and sent to the CO2 storage tanks. That is what it is done usually. That CO2, whatever is captured, will be used for several other purposes. But if you observe here, the permeability and obviously the selectivity and how much affinity it has towards the CO2, um, that is also very important for any membrane whatever we are selecting. And flue gas, whatever we are pumping onto the membrane, should be at high pressure. And these membranes are several membranes will be arranged in a parallel mode, and as CO2 passes across this membrane, it retracts on the shell side of the separator, where the pressure is reduced finally from high pressure to low pressure. Of course, this design is just a very simple design which you can understand, and it works in a steady state process. It has no moving parts, and the membrane separation of CO2 from active hydrocarbons in the flue gas. Some of the industries are adopting this membrane separation process. However, this gas separation membranes cannot be exclusively used for CO2, because flue gas, obviously, as I told you, it requires high pressure, and relatively high mixture flow should be there. So to get all this, again, lot of energy is being consumed in this process. That's why it is, of course, I agree, there is membrane separation process that is being adopted at industry, but it has its own disadvantages. That is what I am telling. And then coming to gas absorption membranes. Previously, what we have seen is gas separation membranes. These are gas absorption membranes. What is the difference between these two? Here also there is a microporous membrane, as you see in this figure, and flue gas stream is passing in a counter current fashion with respect to the absorbent on the other side of the membrane. So CO2 separation is needed. That is our target. So what we have to do is, it should be first diffused across the membrane and then uptaken by a liquid absorbent. This is a liquid which will be taking up the CO2 due to high driving force, because absorbent, it is continuously flowing, right? So lot of concentration difference, lot of driving force for the mass transfer to occur, because the concentration gradient actually drives the mass transfer. Here the absorbent is continuously absorbing the CO2 which is being liquid, and it is flowing continuously. That's why lot of mass transfer occurs, because of the concentration gradient that is occurring, high driving force. This operation leads to higher removal rate than gas membrane separation, what we have discussed earlier. This is a counter current mode of operation. So the development of independent monitors for liquid flows. This is liquid flow is in terms of absorbent, and gas flow is CO2. If we continuously monitor the liquid flow, gas flow, this definitely leads to an efficient operation when compared to a gas membrane separation. So this is all about whatever methods are available for CO2 sequestration. But being biotechnologist, we would like to actually enhance the CO2 capturing technology using a biological way, and the best available source for that is algae. Because I told you already in the previous lecture, algae is a photosynthetic microorganism. It has to take CO2 for its growth. As I have mentioned in my earlier session, second or third slide in a schematic diagram, so CO2 is required for algae to grow. So algae will sequester this CO2 from the atmosphere during photosynthesis, and whatever CO2 is taken from the atmosphere in autotrophic mode, that will be converted as biomass. That biomass can be used as biodiesel, or it can be used as some value added product. Depends on the algae whatever we are choosing. So photosynthesis, as you know, it takes place in chloroplast, by which solar energy is converted into chemical energy, and that chemical energy will be stored in the form of lipids or carbohydrates, which can be further processed for biofuels, foods, animal foods, or other value added products. So the light independent reaction is the one which begins with capturing CO2 molecules. There are two reactions, light dependent and light independent reaction, during the photosynthesis process. In the light independent reaction, PS2 reaction, it begins with capturing CO2 molecules by ribulose 1,5 bisphosphate, RUBP, in short form we'll call it like this. This particular capturing of CO2 by RUBP is catalyzed by Rubisco enzyme, which actually determines the carbon dioxide fixation. So, in the Kelvin Benson cycle, this all happens, and during this step, organic compounds are produced as sugars from CO2 and H2O, with 3 molecules of ATP and 2 molecules of NADPH, which are required to reduce each molecule of CO2. So, if you see this carbon concentrating, you might get a doubt, ma'am. Okay, atmospheric CO2 that will be taken in the light independent reaction in the PS2, which is catalyzed by Rubisco enzyme, because Rubisco enzyme is having the capability of capturing CO2. But what about the dissolved CO2? Because when we operate in open pond systems, especially, the CO2 which is present in the flue gas or which is present in the atmosphere might get dissolved in the medium, whatever in which we are growing our microalgae. Then, how will it be sequestered? That is a common question everyone will be having in their minds. So, here there is a point, like, see, um, when pure CO2, when it is getting dissolved in water, the pH drops. Why pH drops? Because carbonic acid or bicarbonate acids are being formed, which is making the media in which algae is growing, the media is converted into acidic medium, and the equilibrium reactions are written in this way: CO2 plus H2O, H2CO3, again HCO3 minus plus HCS, H plus, again it will give CO3 minus 2 plus 2H. So, but we have understood that Rubisco enzyme is having only capability to capture CO2 but not dissolved CO2. So, thus another enzyme which is called carbonic anhydrase, CA, is used to convert this carbonic and bicarbonate acids into CO2, and then again Rubisco plays its role, and finally it converts the algae into the biomass in the photosynthesis process. So, both carbonic anhydrase in the liquid, whenever CO2 is dissolved, this plays an important role. Rubisco enzyme, when CO2 is present in the gaseous form, it plays a major role. So, both enzymes are found in both micro and macroalgal species, allowing for active and passive carbon capture. What is meant by active carbon capture? If CO2 is present in gaseous form, it is active carbon capture. Passive carbon capture means when it is dissolved. Then also this carbonic anhydrase enzyme is playing an important role to convert it into again CO2, where again Rubisco enzyme comes back into action and sequesters this CO2 by utilizing in the photosynthesis process. So as we have discussed, there are various modes of algal cultivation systems: open pond systems and closed systems. This is a picture which I have taken from Google, and this is the bioreactor bubble column, energy efficient bubble column photobioreactor, which I have designed as part of the DMT project, which is a 15-liter capacity bubble column photobioreactor. So any mode of operation can be used for algae cultivation. However, each mode has its own advantages and disadvantages, which we will not discuss now. It is not of our concern now, but just wanted to show you what are the algal cultivation systems. So, uh, now how to analyze what is CO2 fixation efficiency? Definitely, pure CO2 is not required for algal growth cultures. CO2 can be even sequestered from the flue gases that are being emitted from the combustion process. So, the carbon dioxide capture efficiency mainly depends on what type of algal strain we are using, the concentration of CO2 cultivation system, the environment and the operating conditions, whether it is open pond, closed pond, the pH, the light intensity. There are many factors which we will discuss now. So, the biomass and growth rate measurements will help us in understanding how much CO2 is being fixed during the algae CO2 sequestration. And there is a simple formula to understand this CO2 fixation efficiency, that is CO2 influent gas minus CO2 effluent gas. What is the concentration of CO2 in influent gas minus concentration in effluent gas by CO2 influent gas into 100 will obviously give the removal efficiency of CO2. And fixation rate RCO2 gives us the rate of CO2 fixation in terms of meter cube per hour, gram CO2 that is being fixed per 1 meter cube for 1 hour of algal culture. So, if we analyze that particular formula, it is CCC. Getting this data at one stretch is very, very difficult, because if you really want to understand what is a CO2 fixation, CO2 removal efficiency, you need to refer many, many papers. That's why these kind of sessions will help you get a knowledge at one shot. You can, if you are really interested in this particular field, you can directly use these techniques or use this formula to analyze the CO2 fixation. So here the mule, whatever we have represented in the formula, that indicates the volumetric growth rate in linear growth phase, exponential phase. That means how much grams of dry weight of algal biomass is being produced per 1 meter cube in 1 hour. That is mule. MCO2 and MC, those both represent molar mass of CO2 and elemental carbon. And CC represents in the formula the average carbon content in the biomass, algal biomass that is being produced. How will you estimate the average carbon content in the algal biomass produced? Using elemental analyzer. You can use an elemental analyzer, and there are some standard thumb rules that are already available from our literature studies. That is, for microalgae, average carbon content is measured by an elemental analyzer whenever you do it, because it is a stoichiometric equation which always follows that particular stoichiometric equation. So it is generally observed that 0.507 grams of carbon per gram dry cell weight is being produced. If 1 gram of algal biomass is produced, in that 0.507 grams is carbon. That is a standard thing. You can use it like a thumb rule as such. And also another simplified method is available, that is 1 kg of produced biomass. If 1 kg of algal biomass is produced in your particular system, then you can confidently say that 1.88 to 1.92 kgs. Actually, it's not just 1.88. There are various stoichiometric equations based on the nitrogen source that is being used, but it ranges from 1.88 to 1.92 kgs. That means, suppose if you are growing algae using atmospheric CO2 or from a flue gas in an open pond or a closed system, and if you are able to produce 1 kg of biomass in your cultivation system, then you can confidently say that you have treated or you have recycled 1.88 to 1.9 kgs of CO2. That is a, again, a standard thumb rule that is available. There is a simple calculation that is involved. If you are interested, I'll let you know later. So okay, then coming to algae types, there are as I discussed, there are microalgae, macroalgae. Macroalgae, generally we call seaweeds as the macroalgae. They are marine, obviously, and but they have lot of potential. They have very much good growth rates when compared to terrestrial plants, but obviously their growth rates are less when compared to microalgae. But even marine microalgae also have a very good growth rate when compared to terrestrial plants. But why this macroalgae are not being commercialized for CO2 sequestration means, the only reason what I have pointed out earlier is they have less capability to produce lipid production, which can finally lead to biodiesel. So and again, you need to genetically modify the marine microalgae, and then you have to enhance the lipid production as well. That's why there is no much focus on macroalgae when compared to microalgae. I just wanted to show you some macroalgae pictures. This is macrocystis, this is laminaria, marine microalgae, and this is sargassum, a brown weed. Okay, and this is palmaria. This is also one of the seaweed marine microalgae, which is classified as rhodophyte. There are various kinds of marine microalgae that are available. Even the green colors like vulva, enteromorpha, all these are also having high rates of CO2 capture per gram fresh weight of this particular seaweed, and they are just grown on seashores. Actually, there are many scientific organizations, government scientific organizations, particularly in Goa, they are operating, they are growing marine microalgae. Even some commercial industries are there in Vizag, Ongole, and all, where they are working on marine microalgae. Of course, not for CO2 sequestration, but for some other value added products which they want to extract from this, because they are very easy to grow when compared to microalgae. But however, there are some modifications to be done during their cultivation system and process and all. Then coming to microalgae for carbon dioxide sequestration. As I told you, microalgae obviously have higher potential for lipid content and hence microalgae are concentrated in carbon dioxide sequestration. They are capable of doubling their biomass, depends on the strain, 4 to 6 hours. Some microalgae can double in 6 hours, some in 8 hours, some in 4 hours. So depends on the strain whatever you are taking. And also they can withstand some harsh environments as well. As I told you in the previous session, Hematococcus pluvius can even withstand higher temperatures of 40 to 50 degrees centigrade. That's why they are grown in deserts. So some microalgae can withstand some harsh environments as well, and oil content also can reach as high as 80%. So microalgae can be used as CO2 storage and conversion to fuels. And some commonly used freshwater microalgae are spirulina, chlorella, synodismus, Nanochloropsis. Nanochloropsis is a marine microalgae. Whatever I have mentioned here, all these are marine microalgae, and these are freshwater algae. Research is being done on this particular species. And what are the factors that are affecting the growth of algae? Actually, this is very much related to our previous topic. We have actually optimized the conditions like pH, temperature, light, the light pathway, the irradiance pathway, whatever we call that, also plays a very very important role. The day night cycle, whatever we maintain, all these actually affect the growth of algae. Why we should consider the growth of algae when we are talking about CO2 sequestration? Because here you need to understand this point. Why I am talking about factors that are affecting growth of algae? Because if photosynthesis is more, that means if chlorophyll content that is produced in microalgae is more, then obviously CO2 capturing is more. Those both are directly related. That's why if growth of algae is more, then definitely CO2 sequestration is more. This is an indirect way of assuming the CO2 sequestration. So, light being one of the important parameter in the growth of algae, we'll be supplying in photobioreactors artificial light, and in open ponds the sunlight will be used. Even the photo period, that is 16 is to 8 or 12 is to 12 or day night cycle, all this play a very important role in the growth of algae. 24 hours cycle also will be maintained, where continuously light will be supplied without any dark cycle also. And there are various ranges of light intensities. What I have operated is till 200 micromolar photons per meter square second. That is the standard SI unit for light intensity, micromolar photons per meter square second. But in general, they will be operating from 100 to 200, and saturation light intensities ranges from 140 to 210 micromolar photons per meter square second. Some algae also require low illumination, and some require high illumination. Whatever I have operated is mostly chlorella species only, and it requires approximately 200 micromolar photons per meter square second. Some algae even require 420 millimoles of irradiance, which is the maximum range. Depends on the strain whatever you are taking. You have to experiment, then only you will know. Or a literature can give you a hint of the optimum light intensity for that particular microalgae. And even we have conducted one more study which I didn't highlight in the previous session, but we have operated light intensities in blue and red spectrum as well, to observe how the photosynthesis is carried out in the blue and red spectrums inside the microalgae when they are provided in the photobioreactor. One, we observed that in red and blue lights, chlorophyll A and B contents accumulation inside the microalgae was at its maximum. That was one of the interesting conclusions which we have got from microalgal growth. Then one more parameter that is affecting the microalgal growth is pH. Obviously, pH is very very important in obtaining high biomass productivities. And as I told you, when CO2 is being given as input, obviously there will be lot of fluctuation in pH levels. Finally, of course, the pH will be neutralized, because as we discussed about the Rubisco enzyme and the other enzyme, they both will compensate the CO2 levels, and finally the pH of the medium will be maintained at neutral pH. That is what it is mentioned here in this particular line. Because I discussed earlier, I don't want to discuss because already it is becoming 9 o'clock. Then NOx, SOx, and CO2 from flue gas into algae can also cause the pH to drop significantly if not properly controlled. And then coming to temperatures, in general, microalgae operates better at 20 to 30 degrees centigrade. Um, in general. But some species, as I told you, might operate at even higher temperatures as well. And some microalgae even grows at polar regions, which are grown at very, very low temperatures. But when you are designing a photobioreactor, you need to keep in mind how to control the temperature. We have created a particular strategy to control the temperature in the photobioreactor, whatever we have designed at a 15-liter level. But anyone who is designing a photobioreactor should even concentrate on this particular temperature, because temperature increases during the growth of microalgae because of the light that is being provided, or because of the metabolic activities that is being taking place inside the photobioreactor. So temperature should obviously be controlled, otherwise microalgae will die without any doubt. Then impact of flue gases on algal biomass. So flue gases are considered as worthy carbon sources for algae photosynthesis, but sometimes they contain heavy metals as well. In addition to CO2, as I told you, it contains 142 various compounds, flue gases. When we are operating the microalgal cultivation system with a flue gas, it is very much important to know what is a flue gas composition, and it must be controlled. Suppose heavy metals are there, or some SO2 kind of toxic compounds are there, first they must be treated, and then only flue gas can be given as an input into the algal cultivation system. Otherwise the algae cannot tolerate these toxic compounds. Uh, of course, NO can be metabolized by algae, but not SO2. Okay, so we need to analyze that, only comes with experience. And finally, I would like to conclude, this is our model which we have developed for CO2 sequestration using microalgae. This is a 50-liter scale bioscrubber, 20-liter scale bioscrubber. Um Inside this we have an open tank which is pumping the algae in these tubes in this mode. And this is the second day, this is seventh day, and this is 12th day of growth of microalgae. And we have exposed it just to the open air. Of course, here we have covered it, but it is open tank, and sunlight is being provided continuously. Natural sunlight. We are not giving any artificial light, and this is what we claim. Um, Irrespective of the season or irrespective of the strain we are operating, we assumed that if 1 gram per liter of biomass is produced, then as per the standard thumb rules, whatever I have pointed out earlier in the previous slides, we assume that 2 grams of CO2 is being absorbed for every 1 gram of biomass being produced, 50% more. Okay, so if 20 liters, in 20 liters, for 1 liter, if we get 1 gram, 20 liters will get 20 grams. So 40 grams of CO2 is being absorbed per day. That is our conclusion. And if we take it for year, this is the value. And grams per year in kilos, if we take, it is 14.6 kgs of CO2 is being sequestered in our 20 liters capacity bioscrubber. But we have collaborated with a company called Neon Flakes for this particular project, where I am the consultant actually for that particular project of bioscrubber development, where we are developing this 200-liter capacity, where our final goal is to sequester 219 kgs of uh, CO2 for the 200 liters capacity whatever we are taking. That's what I am pointing out. Our target is to increase the biomass to at least 1.5 grams when compared to 1 gram at 20 liter capacity, so that we can increase the value at 200 liters capacity. And when you compare these values with our CO2, the trees which they are observing 70, current machine it is approximately 60 grams in a day it will absorb, and the future scaled up bioscrubber might absorb up to 600 grams. And this is our scaled up this is the picture which we are designing the 200-liter capacity bioscrubber, and here we would like to even integrate the wastewater treatment to reduce the costs that are involved in the artificial medium. We also propose to use the algal biomass that is being harvested from this particular tank to be used as a biofertilizer, which in turn supports the biocircular economy. We are taking CO2 from atmosphere, we are taking wastewater from the ponds or nearby polluted lakes and all. Then we are treating that. Then we are obtaining algal biomass. That algal biomass is again being sent into the earth in the form of biofertilizer. So it completes the cycle, which we claim it as biocircular economy. And this is all about CO2 sequestration, what I wanted to talk about. Thank you so much for this opportunity, and the session is open for the doubts.
Thank you very much, ma'am. Fantastic, presentation.
Thank you, sir.
So especially that the carbon sequestration, is one of the hardcore area where we need to address that the social impact of the CO2, and that also is contributed to the global warming. So this is an upcoming area, and where we can able to go for the funding opportunities also. So
Yes,
right
sir.
time you are projected that the algal, biomass, which help to absorb the or capture the CO2, so that it is
Yes.
reduced in percentile aspects. So thank you
Yes.
very much, ma'am.
Thank you, sir.
Uh,
Actually, in India, there are no commercial systems that are available for this, sir, with algae. So, if anyone is interested, they can do some research on this, and they can definitely propose some for some funding opportunities as well. If at all
yeah.
participants are interested, because most of them will be faculty. That's why I just wanted to point out this.
Yes, ma'am. Yes, ma'am. Yes, ma'am. Thank
Yeah.
you. So participants, if you have any queries on your mind, so feel free to unmute, and you can able to discuss with the ma'am. Ma'am, I have one question
Sir.
about the algal biomass. In case of the like bubble column that you are developing for the algal biomass, right? So instead of the bubble column, so can we go with some kind of airlift reactor for the development of algal biomass?
Yes, sir. Definitely. It's not that we have to restrict ourselves with the bubble column bioreactor. Airlift bioreactor also we have operated, bubble column and CSTR also we have operated. Airlift has its own advantages when compared to bubble column reactor. But the bubble column reactor, what we have designed, has internal illumination, sir. That design I'll discuss with you whenever we have some free time.
Yes,
That
yes.
internal illumination, whatever we have kept inside the reactor, will act as baffles inside the reactor, which will increase the turbulence, which will increase the mass transfer, which will in
Okay.
turn increase the CO2. We have opted bubble column bioreactor, but it's not a hard and fast rule that we have to only go with bubble column. We can go with
Okay.
any type of reactor. Mm.
Okay, ma'am, is it any mixing or agitator can be provided in
No, sir.
the bubble column
And
reactor?
that's why we are claiming as energy efficient. No moving parts, sir. No moving parts, absolutely. Only because of the baffles, whatever we are claiming as
Okay.
inside the bioreactor, that will act as the agitator. Simply,
Okay.
I'll show you the
Okay.
design, sir. Once we'll
Yeah,
discuss
definitely, ma'am.
about this. Yeah.
I think Arun Prasad sir, you want to discuss with the ma'am, so you can unmute
Ma'am,
yourself. Yeah. Yep.
good evening, ma'am.
Good
The
evening.
was very nice.
Thank you, sir.
So now we are searching in third generation biofuel in converted into oil. So we are using in IC engine also. So I have one doubt, ma'am.
Yeah.
Compared to marine algae, fresh algae, which one is the most oil content?
Sir, fresh algae, sir, we can't actually compare like that. But whatever we are operating in our local condition, marine algae we can't grow in Hyderabad, sir. So we are only operating on fresh algae. But in general, marine algae is also being used, especially the nanochlorosis, whatever I have highlighted. We are creating artificial medium, the marine medium, and we are using that. But nanochlorosis is claimed to have more lipid content when compared to other freshwater algal species as per the research reports I am talking.
Okay. Thank you very much.
Yeah.
Ma'am, Divya ma'am, do you have any concern?
Yes, sir. Am I audible, ma'am?
Yes,
Yes.
ma'am. Yes, ma'am.
Ma'am, first of all, I have to thank for Tech University for arranging this event. So, and your presentation is also too good. Actually, I'm from the major of ME Environmental Engineering. The session is very interesting, and I have a small doubt regarding my thesis also, ma'am.
Yeah.
In my research work, I am using the algae that is called coccolithophore kind of algae. So, do you have any idea about it, coccolithophore?
No,
It
is
is
it?
also a
I
kind of.
never heard actually. Is it a fresh algae or marine algae?
It is a marine algae, ma'am. It is available in marine algae, which is capable of producing lime too. So I have more doubts on that particular thing. So I have, because you have said about marine and actually these algae are also grown in open pond as well as the baffle reactor. So I have raised this query. So did you hear about this kind of algae or not?
No, but one thing we can do, we can have a discussion over an email. You can mail me if you have any particular doubt. I can help you if I have some knowledge about that. Yeah.
Okay, okay.
I will share with you, ma'am, ma'am email ID and contact number. However, the
Yes, sir.
it is there in the the schedule which I shared. Before commencement of our FDP. So
Yes, sir. I have a
you just
poster.
scroll down. Yeah. You can drop an email to ma'am. So definitely, ma'am will address.
Sure. I'll
Okay.
be happy to help you in your PhD thesis also, Divya. Let's discuss on that. Yeah.
Yeah, okay. Thank you so much, ma'am.
Okay.
Any other queries on your mind? Otherwise, we will go for the vote of thanks. Yeah, comparative, you can go for the.
Yes, sir. Thank you, ma'am, for sharing your valuable insights with us. Gratitude is not only the greatest of virtues, but the parent of all the others. With this, I would like to call Dr. A. Sarvanaraj, Deputy Dean, to give away the vote of thanks.
Good evening. Hope I'm audible.
Yes, sir. Yes, sir. Please go ahead, sir.
Yes, sir. On behalf of the Biotechnology and Chemical Engineering Department, Tech High Tech Dr. Rangaraj and Dr. Sagantala Engineering College, it is my pleasure to deliver the vote of thanks for the third day of our ATAL FDP on Waste to Wealth, Emerging Trends in Green Energy for Sustainable Development. Our sincere thanks to Dr. Rajeshri ma'am, Associate Professor and Head of Biotechnology, Chaitanya Bharati Institute of Technology, Hyderabad, for having accepted our request to deliver the lecture on and CO2 sequestration as well. With gratitude, we would like to appreciate the efforts taken by you, ma'am. Thanks for your valuable time spent with us. As you have rightly pointed out, for knowing specific information to carry out any research activity, it is so hectic to refer so many published articles and materials. Your simplified and extraordinarily informative presentation has enlightened us, all of us, of course, in the topics of CO2 sequestration and, of course, energy and exchange analysis. Thank you so much once again, ma'am. Hope we will continue our association in future as well, in terms of sharing knowledge and in terms of research. Thank you so much, ma'am.
Thank you so much, sir, for your wonderful words.
Yes, ma'am. It's our pleasure.
Yeah. Thank you, Dr. Gopinath, sir, for giving me this opportunity.
Yeah,
actually.
Thank you, lots, ma'am. We'll work on it, ma'am. Future projects also.
Sure, sir. Sure. I'll be happy to collaborate.
Yeah, definitely. Thank you.
We wanted to thank all the participants for your involvement and your effective participation. We are conducting this FDP in online mode for these 6 days till Saturday, and from Monday onwards it will be offline. And we feel that it will be a good get together for all of us. Of course, we are planning for an industrial visit. We are informing you earlier today itself by Monday. So I think we'll cherish this FDP forever. Thank you so much for all your support for all the participants. Thank you so much.
Thank you, sir. Thank you very much, sir. Ma'am, thank you very much, ma'am, once again on behalf of the entire team and Welltech fraternity. So we'll keep work on it, ma'am. Thank you. Bye-bye.
Excellent efforts by your team, sir.
Yes, ma'am. Yes.
All the best.
Thank you, ma'am. Thank you, lot.
Okay, sir.
Thank you, participants. Feedback and the attendance link is posted in the chat room, so you can request it to fill out your attendance and the feedback. So we'll meet tomorrow, for the 4th day of session. So once again, thank you all. Good night. Bye bye. Comparenting, thank you very much. Pavitran, thank you. So you can sign off the YouTube link. Hi, sir. Thank you very much.